Thank you all for coming. Now presenting Vlad Vitok with MAIA Biotechnology Incorporation.
Thank you all for coming. I'm the founder and CEO of MAIA Biotechnology. It's a pleasure to be here today to represent the company. MAIA is an extraordinary company. We are the makers of the first telomere targeting agent program. THIO, our lead compound, is in the clinic. It's in a phase 2 go-to-market clinical trial. And the efficacy that we have seen with THIO in different models of cancer is absolutely outstanding. We have seen curative effects at doses 40 times under the maximum tolerated dose. Such a package of efficacy and safety is unprecedented. The FDA have reviewed the data of THIO and granted THIO 2 orphan drug designations of the first 2 applications. REGENERON as well, via the clinical supply agreement, participate with their drug LIP-THIO, an immune checkpoint inhibitor, into the phase 2 THIO-101 trial in non-small cell lung cancer that is currently underway, enrolling in Australia and Europe, and on track to open sites in US later this year. The upcoming milestones from this trial are near the safety we already published. We're going to have overall response rates coming through in Q4 and duration of response next year. A second phase 2 go-to-market trial called THIO-102 is also set to start later this year, and the third one, THIO-103, in first line of therapy as well. Here's a look at our robust pipeline. Up top, you have the THIO telomere targeting agent trials. THIO-101 in non-small cell lung cancer. We're testing THIO in sequential combination with LIP-THIO of REGENERON. It has begun enrollment in July of last year. Enrollment is going very well. THIO-102 is a basket design with 3 cohorts. Colorectal, HCC, stands for hepatocellular carcinoma. This is the dominant histology in the primary liver cancers, about 90% of them. Then solid tumors of all types as a signal generation arm. Here we're testing THIO in sequential combination with 3 different checkpoint inhibitors to start later this year. THIO-103 is a confirmatory phase 2/3 trial in non-small cell lung cancer and small cell lung cancer in first line. This trial is designed to confirm the results from the other 2 trials, which in turn are for accelerated approval. The second generation of telomere targeting agents is a program well underway. The first 2 are now in development, in preclinical development, and are expected to go to clinic in the second half of next year. So THIO is short for 6-THIO-DG or 6-THIO-deoxyguanosine. It is a telomere targeting agent, the first. A small molecule that penetrates the blood-brain barrier, capable of treating brain cancers, eligible for new chemical entity, NCE, marketing exclusivity, the strongest you can get in the US and in other countries as well, has a dual mechanism of action. Telomere targeting and immunogenic effect turns immunologically cold tumors into hot and responsive to checkpoint inhibitors. We have seen complete response with no recurrence in multiple tumor types, and the FDA orphan drug designations came for HCC, hepatocellular carcinoma, and for small cell lung cancer. The next generation is a program we started about a year ago, and they are all telomere targeting agents. They work via a similar mechanism of action as THIO. Some structurally are an evolution of THIO, and others are new structures. We tested them, or we developed 84 new molecules. We tested them in vitro and then in vivo, and 7 of them proved to be an order of magnitude superior to the original in efficacy in certain tumor types. And the objective here is to advance to development 1 agent every 12 months. The first 2 started already. So cancer, unfortunately, is a problem that is here to stay. It's not going anywhere anytime soon. It's the most dominant age-related disease, and it affects the elderly, in particular, people north of 70 years of age. The population aged over 80 is expected to triple in the next couple of decades. And at age 90, the probability to be diagnosed with cancer, with a type of cancer, is 40%, and to die of it, 20%. By age 100, these numbers more than double. It is a function of aging. THIO is the only direct telomere targeting agent currently in clinical development and has a dual mechanism of action, telomere targeting and immunogenic effect. So first, telomere targeting. Here you have the chromosome to the left, depicted in blue, that contains our DNA, our genetic information. And at the end of the chromosome arms are the telomeres, our target, here represented in orange. The telomeres have a very specific function to maintain the integrity of the chromosome in the course of the cell division cycle. They are built and maintained by the enzyme telomerase. Telomerase is present in all normal cells in the first year of life, but at about age 1, it goes away. At that point, the telomeres have reached their maximum length, and from then on, with each cell division cycle, they lose a little until in the old age, they become critically short, can no longer protect the chromosome, and mutations begin to appear. With this, the diseases of the old age, including cancer. But in cancer cells, something extraordinary happens. The enzyme telomerase is turned back on. And so the cancer cells regain an ability to elongate their telomeres and reach a state of replicative immortality, a scientific way of saying they continue to divide and the tumor grows and grows in the aging body. And this is where THIO comes in. THIO is picked up by telomerase, placed in the structure of the telomere, creates a faulty structure. The telomere collapses, the DNA unwinds, and the cancer cell dies. So the enzyme telomerase picks up THIO, places THIO in the structure of the telomere, creates a faulty structure. The telomere collapses. The DNA unwinds, and the cancer cell dies. And this process is fast and efficient. It happens in 24 to 72 hours, and THIO directly kills 70 to 90% of the cancer cells. Then follows the immunogenic effect. Of the telomeric fragments, THIO forms micronuclei and then presents the telomeric fragments to the immune cells, triggering an immune response so effective that if you follow THIO with an immune therapy checkpoint inhibitor, you see complete response, no recurrence, and anti-cancer immune memory. In effect, THIO teaches the immune cells to find and kill the cancer cells on their own without any additional therapy. We have seen this happening with Libtayo of REGENERON, with Keytruda of Merck, with Tecentriq of Genentech, in many different tumor types. And the first go-to-market is in non-small cell lung cancer with Libtayo of REGENERON. So a few words about the REGENERON partnership. This is in the format of a clinical supply agreement. Under this, REGENERON provides their drug Libtayo to the THIO-101 phase 2 trial in non-small cell lung cancer, in which we test our THIO with REGENERON's Libtayo, cemiplimab. This is a very big deal for us. It's 184 patients times 12 months of therapy times $15,000 per month. It's a $32 million participation. Separately, we raised $32.4 million, well, now almost $40 million, by selling shares. But this participation is non-dilutive. REGENERON do not take a position in the cap table. All they take is development exclusivity in non-small cell lung cancer for the duration of the trial, which is estimated at 2 years. This means we cannot do a trial in non-small cell lung cancer with a direct competitor of LIBTAYO, another anti-PD-1 agent. Any other combination remains open in non-small cell lung cancer, and in any other tumor type remains open as well. It's a very good deal. Our best financing move today. A few words about THIO-101. This is the ongoing trial that started in July of last year in Australia and now is enrolling in Europe as well. It is testing THIO administered in sequence with LIBTAYO, cemiplimab, in non-small cell lung cancer, advanced non-small cell lung cancer, in second line of therapy. So, unfortunately, most patients diagnosed with non-small cell lung cancer present with advanced disease, which is inoperable and incurable with current treatments. They are typically treated with KEYTRUDA plus minus chemotherapy in first line. But after 5-6 months, they progress. When they progress, they come to our trial. In our trial, we are testing 3 doses of THIO, 60 milligrams per cycle, 180, and 360. 60 milligrams per cycle is the curative dose in mouse translated to humans. Compare this to the maximum tolerated dose of 2,500 milligrams per cycle. It is 40 times under. Unprecedented. Now, we are testing higher doses as well, just to make sure we do not leave on the table any possible additional efficacy. But even the highest dose, 360, is still 7 times under the maximum tolerated dose. So for the part A of the study, safety lead-in, we started with the highest dose, 360. The way we give it is 3 consecutive days. We break the dose in 3, 120 milligrams per day, Monday, Tuesday, Wednesday. It's an IV, easy to take, 15 to 30 minutes, just like LIBTAYO is. Then 1 day of break on Thursday for the immune activation. And then LIBTAYO, cemiplimab, on Friday. Dose as approved. We don't change anything about LIBTAYO. We just bring THIO prior to LIBTAYO to make it work a lot better. The cycles are every 3 weeks. We completed part A, safety lead-in. There are no issues there. And now started part B, and it's enrolling very fast in part B. The objective here is to select the best dose of the 3 on the basis of overall response rates and duration of response, and then expand that into a registration size arm for accelerated approval in US and Europe and go to market. So the safety profile from part A, very, very favorable. We tested the highest dose, 360. There was no dose-limiting toxicity, no serious adverse events. The safety profile is substantially better than the current standard of care, which is chemotherapy for second line, and has 70 to 80% incidence of grade 3 and 4 very severe side effects that require stopping treatment and oftentimes hospitalization and sometimes even death. So nothing like that here. So the Safety Review Committee recommended the start of Part B, the efficacy and dose selection, and we have done it. We started it. So here's a look at the safety profile. What have we seen to date? For those unfamiliar, in oncology, safety is graded from 0 to 5. 0 is nothing at all. 5 is dead. We have seen grade 1 adverse events, very mild. Fatigue, decreased appetite, blood pressure fluctuation, dyspnea, nausea, basically describing how I wake up in the morning. There was one incident of grade 2, one of grade 3, and they were resolved very quickly, and so no dose-limiting toxicity. But what's even more interesting, we also already see the preliminary survival results. In the first 2 patients enrolled in Part A in July, they continue to be alive after 10 and 9 months after beginning of treatment. This is extraordinary. Both patients were advanced stage 4 disease, heavily pretreated. They went through 3 and 4 lines of treatment. They tried everything before coming to this trial. They progressed through all of them. They came here, they were treated, and they are now without progression several months out, 7 and 6 months out, respectively. This is unprecedented. In this type of late line of therapy, with therapy, patients barely survive 3 and 4 months. Without therapy, they die in weeks. This is the preliminary survival data in a chart. You have up top the 2 patients. We are tracking all of these patients as we go. You see in the stars represent the cycles of therapy when they were administered. This is amazing data so far. Since this chart was created, everybody progressed and they got another cycle more, plus more patients were added. So it's running very well. Now, the reason why the MAIA and REGENERON teams believe this is a high probability of success trial is that we have seen extraordinary results in models of lung cancer. This test was conducted in 2019 and published in 2020 in Cancer Cell, a very reputable journal. Only the best quality data makes it in. We tested THIO in sequential combination with Tecentriq, atezolizumab of Roche and Genentech. And the results of this were the basis under which the agreement with REGENERON was started. So the results we plot in this graph, tumor volume on the vertical, and then days after tumor inoculation on the horizontal. Only 2 cycles of therapy were administered in the very beginning of the trial. So we see the control arm in gray, without therapy, grows fast. This is a tumor type that's very aggressive. Atezolizumab, Tecentriq, as monotherapy in orange, grows just as fast. This is not, it's an immunologically cold tumor. It does not respond to immune therapy by themselves. In green, you see THIO monotherapy with a solid effect. It blocks the tumor growth for some time. But in blue, the sequential administration of THIO and Tecentriq with an extraordinary result. Look at the delta between the curves, blue and gray. 60% complete response. To put things in perspective, Keytruda, the market leader in the clinic in non-small cell lung cancer in first line, has 1% complete response. This is 60. We later repeated this with THIO followed by Keytruda and followed by Libtayo. Similar results. Then we follow the complete responses for 70 days. There is no recurrence. 70 days in mouse are like 7 years in humans. The curative threshold is 5. So at this point, they are cured. But we go above and beyond and re-challenge with implanting 5 times more lung cancer cells and with no additional therapy. There is no take. Immune memory is in place because of THIO. This is beyond cure. So with this data, we go to non-small cell lung cancer. Non-small cell lung cancer second line of therapy, where the current standard of care is chemotherapy. Very toxic, not effective. Response rates published are 11 to 23%. We talk to our investigators, they say, yeah, that is literature, but in real practice, we don't see double-digit response rates with chemo, just single digits. There is no complete response to talk about. The duration of response, progression-free survival, very short, 4 months. Survival up to 10. It's just very bad. Any 20% improvement on these numbers makes the drug approvable. 30% response rate combined with a 6 months or better duration of response is approval in an accelerated manner. Very high probability of success trials here, indeed. So we have a biomarker of efficacy, very reliable. It's called TIPS. TIPS is an acronym that stands for telomere dysfunction-induced foci. These are the yellow dots that appear on confocal microscopy when THIO hits the telomeres and destroys the telomeres. And they maximize at 48 hours after administration. You can see the range is between 24 and 72, and their formation indicates the on-target mechanism of action of THIO. THIO-102, the next trial that is planned. It's a basket and umbrella design, a very modern design. The FDA appreciates this a lot. We start with 3 baskets: colorectal, HCC, hepatocellular carcinoma, following up on one of the orphan drug designations, and solid tumors of any type, a signal generation arm. As soon as we see responses forming around a certain tumor type, we then make it into its own arm for another shot on goal. In each of them, we test THIO in sequential combination with 3 different checkpoint inhibitors: Tecentriq, Libtayo, and Keytruda. Select the best and then expand that arm to become a pivotal size arm for accelerated approval. And in colorectal, we have seen our best results to date. 100% complete response. No recurrence. Even when re-challenged with 10 times more, this time 10 times more colorectal cancer cells with no additional therapy. There is no take. A new memory is in place because of THIO. Colorectal, the situation is even worse. In salvage colorectal, where we are going with this, chemotherapy is the standard of care, very toxic, and it just does not work. Response rates are 2% or less. Duration of response, progression-free survival, 2 months. Survival, 7 months. Any 20% improvement on these numbers is approvable. Another high probability of success trial. We saw similar things in small cell lung cancer, THIO plus Pembro, in HCC, THIO with Atezo. These were the 2 data sets reviewed by the FDA when they awarded THIO the orphan drug designation. And last week, we published the most recent results. This was in HCC, in hepatocellular carcinoma. THIO, by itself, monotherapy, 100% complete response in HCC. As monotherapy, this is where THIO works best. But when you use it in combination with a checkpoint inhibitor, and here we use it with Libtayo, this response is very, very well maintained over time. Even 90 days out, it's still complete response across the board. That's like 9 years. And on re-challenge, immune memory is always there. This is the THIO-103 trial, the confirmatory trial that we're planning. It's phase 2/3. It starts as a phase 2. We start with small cell lung cancer, following up on the other orphan drug designation, and then non-small cell lung cancer first line in combination with Libtayo as well. So as we're running out of time, I just want to take the audience to the last of the slides, which is the multiple value driving milestones that are ahead of us. So first is the IND filing to bring the trial to the United States later this summer. and open other trials in the U.S. as well. We have many investigator-sponsored trials in the works as we speak. Then, overall response rates in Q4 this year from THIO-101, a major inflection point. This has been a multiple evaluation increase inflection point historically. We start THIO-102 and then 103, and middle of next year, we should have duration of response and progression-free survival from THIO-101. Duration of response combined with overall response rates makes the approvable package. And then we follow through with overall survival, and we're tracking so far to file for accelerated approval in the U.S. by 2025. In parallel, we developed the next generation of telomere targeting agents. The first one tracks to 4 or 5 years to commercial behind THIO. We are building a franchise. And with this, I would like to stop here and ask if there are any questions. No time to. Thank you, and I'm available to take more questions just outside the hall here or thank you for the